Welcome to part 2 of You and Your Kidney, human osmoregulation. This next set of slides are all about the last two functions of the nephron, concentration and then secretion. And you'll also get the first two hormones of the course. Lucky you. Just to review about the nephron that you see on the left. We started with filtration that occurs within the Bowman's capsule, where blood, smaller chemicals within the blood and water are filtered within Bowman's capsule. We went through the process of reabsorption, where the majority of material that was filtered in Bowman's capsule is reabsorbed back into the blood. In this lecture, we'll talk about concentration and the removal of water and salts from the filtrate. Finally, we'll discuss what occurs within the distal convoluted tubules, secretion, in which materials from the blood are added back into the filtrate for removal via the collecting ducts. But before we get into concentration, let's talk about nitrogenous waste, shall we? The breakdown of amino acids and other nitrogenous compounds results in the isolation of an amino group. That's NH2. Amino groups in water quickly pick up an extra hydrogen and become NH3 or ammonia. For those of you who are spending your isolation cleaning your house, utilizing lots and lots of ammonia to scrub it down, you should realize that ammonia is really nasty, very toxic material. If you look down at the figure at the bottom, you'll see that ammonia can be utilized as a nitrogenous waste product, but only in freshwater fish. It takes a lot of water to rinse out, flush out ammonia so that ammonia does not hurt cells. By simply taking ammonia and converting it into a less toxic material that still requires some water to flush it out, is the most common nitrogenous waste among the mammals. That is urea. So urea is a simple compound utilized by humans, among other animals. It still needs a little bit of water to flush it out. It is not as toxic as ammonia, but it's still pretty toxic. And those of you who have dogs and a grass backyard understand what I mean by that. There are some animals, mostly reptiles, birds, and insects, that take a step further and convert urea to a drier compound called uric acid. Uric acid is not very toxic at all. Uric acid is also a compound that requires very little water, but does require energy to make. You've encountered uric acid every day when you park under a tree and some nasty birds pellet it with uric acid crystals. Humans can produce uric acid. Most of the time we get rid of it in our urine. Sometimes it collects enough within our joints to cause a painful condition known as gout. So mammals utilize urea as that kind of middle ground where it takes a little bit of energy to make. It is a little toxic, but not as toxic as ammonia, nor as high energetic production as uric acid. Welcome to the world of concentration. This occurs in the loop of Henle. If you remember from previous slides, the loop of Henle descends deep and deep into the medulla, and the medulla is where a very salty osmotic gradient occurs. The function of the osmotic gradient is to drive water out of the descending loop via osmosis. There are no water pumps. Water has to move passively via osmosis. So the function of the loop of Henle is kind of reabsorption of water, reabsorption of sodium chloride in the ascending loop. Let's look at these loops more carefully. Okay, so you can really divide the loop of Henle into two separate structures. The descending loop, and on the next slide will be the ascending loop. Because really, not only are there different functions within each half of the loop, but also the cells are uniquely different. In the descending loop, good test question, there is no movement of salt because there are no salt pumps or salt channels. And when I mean by salt, I should say sodium and or chloride. Instead, the loop of Henle is filled with aquaporins. These are water channels that allow the movement of water via osmosis. Because of the situation that the loop of Henle and the filtrate within the loop of Henle is descending deeper and deeper into the medulla, encountering saltier and saltier neighboring tissues. Water could move by osmosis, right? Low salt to high salt, without any energy. After the hairpin turn, the loop of Henle climbs and ascends towards the distal convoluted tubules. So this is the ascending loop of Henle. Now the functions change. The function of the ascending loop is movement of sodium. Sodium moves in two ways, diffusion and active transport. In the bottommost third of the ascending loop, sodium can move via diffusion. All you need is a sodium channel. As long as there is more sodium in the filtrate than in the surrounding tissues, sodium can move out passively. However, in the upper 2/3, this situation changes. Now there's less sodium within the filtrate and more sodium within the surrounding tissues, which means you're going to have to move sodium against a gradient. This requires sodium pumps. This requires active transport. This requires ATP. So the ascending loop pumps or diffuses out sodium. What about water? Water cannot move in or out of the filtrate in the ascending loop, for a couple of reasons. One, there are no aquaporins present. There are no water channels. So there's no way to move with water. In the descending loop, the gaps between two cells are rather loose, so water can also move out that way. However, in the ascending loop, the cells that line it are glued together via tight junctions. Thus, the ascending loop, there is a physical barrier for water to move in or out via osmosis. First question: what is it that maintains that concentrated osmotic gradient in the medulla? Welcome to the world of counter current flow. If you remember from previous slides that the loop of Henle especially, there's a blood capillary system that parallels the ascending and descending loops. This is known as the vasa recta. If you look at the velocity and the direction that blood moves vis-a-vis the loop of Henle itself, it is moving in an opposite direction or counter direction, hence the term countercurrent. It is also moving at the same rate as the filtrate. The countercurrent system, which is a system of diffusion, is present to maintain the osmotic gradient of the medulla. Okay, this is the countercurrent flow, countercurrent system. As the blood travels down, following the ascending loop, but in this case, traveling down, it is picking up salts released from the ascending loop, as well as salts, in this case urea, from the collecting ducts. After the hairpin turn, the vasa recta then flows in a counter direction away from the descending loop. In this case, it will start picking up more water. The end result is, despite the fact that water and salts are flowing in and out of this tissue, this countercurrent exchanger is maintaining the osmotic gradient within the medulla. This slide is showing you the movement of material and the movement and these concentrations of salts within the medulla. Remember, the cortex is isotonic, has approximately the same osmolarity in the tissues as in the blood, as in the filtrate. But as it descends into the saltiest tissue in your body, the medulla, the surrounding tissues are incredibly salty. In the descending loop, this allows the movement of water to flow from filtrate into tissue, into the vasa recta. Remember that countercurrent capillary system. In the ascending loop, it's already too salty for sodium chloride, so it has to be pumped out at least the last 2/3. However, the first third of the ascending loop sodium can be diffused out. No energy required. Again, this is showing you the vasa recta. As the blood flow occurs, it is picking up salts and then releasing salts, or actually gaining water as it moves into and out of the medulla. Okay, so again, the counter current exchange using the vasa recta function to maintain the osmotic gradient within the medulla. If you were to stop the vasa recta from flowing, you would disrupt the osmotic gradient, which would disrupt concentration within the loop of Henle and result in failure of the kidney to reabsorb water and sodium. The second question I asked a few slides ago was, okay, so we know the countercurrent flow, the vasa recta, is there to maintain the gradient. How is it possible that as you increase the gradient, you can still move sodium high concentration to low concentration via diffusion, energy savings? Okay. Welcome to the world of urea. Now, when I talk about an osmotic gradient, and I stupidly use the word, quote, salt, unquote, what I'm talking about is any compound that can add to an osmotic gradient. The majority, of course, is sodium and chloride, but you can also use things like amino acids, glucose, potassium. Oh yes, and another salt we don't think about as salt, urea. Okay? So, in the inner medulla, the lower part of the medulla, there is a reabsorption of urea from the collecting ducts back into the tissue. Function: urea is used to replace sodium chloride and yet still maintain a higher gradient to allow water to move out via osmosis. So, the effects of urea replacing sodium chloride in the inner medulla is save energy. Okay, let's look on the slide on the right. What you'll see is a cross section of the cortex, the upper part of the medulla, the outer medulla, and the inner part or lower medulla, and the concentrations of salts. Take a look at the concentrations of sodium chloride from the cortex all the way to the outer medulla, and you see that it is actually producing the majority of the osmotic gradient. However, in the inner medulla, thanks to the reabsorption of urea from filtrate back into the tissues within the collecting ducts, you'll see a huge increase in the concentration of urea. This provides two things. Number one, it increases the osmotic gradient, which on the descending loop provides a wonderful gradient for water to move from low salt to high salt. Second point, the increase in urea in the inner medulla allows sodium not to grow, not to increase, maintain a level, basically a constant level as seen in the outer medulla, so that sodium within the filtrate is actually higher than the surrounding tissues. Thus, sodium moves in the filtrate from higher concentration of sodium to lower concentration of sodium within the medulla. Urea is meaningless in terms of sodium diffusion. Again, end result is descending loop of Henle allows for water transport throughout the entire descending loop. And at least the first third within the inner medulla allows for the movement of sodium out of the filtrate, sodium reabsorption, energy free via diffusion. Whew. Concentration was fun, wasn't it? Let's talk about the fourth phenomenon that occurs within the nephron, and that is secretion, or movement of material from blood back into filtrate for removal. Okay, so this slide just kind of shows up how to get rid of things. All right, we start with filtration, but then most of it's return. We go through in the reabsorption process. In concentration, we reserve four more things. Okay. Secretion is the reabsorption. How's that? The placing back of material in the blood back into the filtrate within the distal convoluted tubules before it goes out via the urine. Again, just showing you that secretion can be defined as the reverse reabsorption. We are placing material back into the filtrate. So what gets moved here? The most famous one is taking large material, large not protein material, and putting it into the urine. The most famous, of course, are drug metabolites that utilize some fancy transporters and ATP to get moved into the distal convoluted tubule. So when you're applying for that job and they want you to take a urine drug test, blame your distal convoluted tubules. But drug metabolites aren't the only compounds moved within the distal convoluted tubules. There's also excess potassium in the blood that can be secreted back into the filtrate, again in the distal convoluted tubules. So again, here is the reabsorption and secretion parts for potassium. Finally, the other interesting ion that is secreted is hydrogen ion, hydrogen protons that can be secreted back in if blood pH is too acidic. So, another function of the nephron is to help regulate pH of the blood, primarily by removing hydrogen ions. Okay, so we spent most of part one and all of part one and most of part two talking about the nephron, what goes on in the nephron. Now let's talk about fine tuning. This is using two interesting hormones that affect water and sodium reabsorption. Ready for the first one? Drum roll, please. Welcome to the first hormone, well, non-cytokine hormone of the course, antidiuretic hormone, also known as ADH, also known as vasopressin, because one of the indirect functions of antidiuretic hormone is to increase blood pressure. So you're pressing on your vasopressin. Antidiuretic hormone is a posterior pituitary hormone, and we'll talk about the posterior pituitary, in a couple of lectures from now. Right. Let's talk about the function of ADH. ADH is involved in reabsorption of water from filtrate back into the blood within the distal convoluted tubules and the collecting ducts. The cells here have receptors for ADH. ADH, as the name implies, is an antidiuretic hormone. A diuretic agent is something that allows you to decrease the amount of water that's reabsorbed. In other words, you pee a lot more water than you should. So an antidiuretic hormone is a way to remove more water. It works, it is believed to work rather strangely. I won't get involved in it, but it involves binding to receptors on the cells of the distal convoluted tubules and the collecting ducts, altering the cytoskeletal structure of those cells, and allowing water to move out passively. So ADH is released by too much salt in the blood. Again, ADH is released by the posterior pituitary. We'll talk about the relationship between the hypothalamus and the posterior pituitary in a couple of lectures. And again, it is there to increase water flow back from filtrate back into the blood, to reduce salinity in the blood, but at the same time will also increase blood pressure. Again, this is just simply a flow diagram of the effects of ADH. And this is simply a table showing some of the effects. But I want to talk about the effects of ADH, the effects of the fine-tuning of water absorption on alcoholism. I shouldn't say alcoholism. I should say a Saturday night of binging alcohol. Right? You drink a little too many shots of pick a beverage, and you wake up with that wonderful, wonderful cotton-mouthy, dehydrated feeling known as a hangover. Blame antidiuretic hormone. One of the functions of alcohol is to inhibit the release of antidiuretic hormone. No antidiuretic hormone to provide more water retention. Therefore, there's more water in the filtrate, more water in your urine. And for those of you who remember or not, the effects of tequila shots, rum shots, whatever shots, is a frequent use of the restroom. So again, it's because ADH is no longer effective. Because you're peeing out more water, you wake up dehydrated. Yes, the hangover. So, if you want to still enjoy getting drunk, make sure with every shot of whatever alcohol that you're taking, vodka, that you also drink a big glass of water in between. And that should, you go to the bathroom a lot, but it should avoid the wonderful hangover feeling. The last part of the lecture and the last hormone that we're going to talk about is aldosterone. Aldosterone is a steroid hormone produced by cholesterol. It is produced and released by the endocrine gland, the adrenal cortex. The effect of aldosterone is to control sodium levels in the blood. Thus, when sodium levels drop, the adrenal cortex will kick in and will release aldosterone. Aldosterone works also at the distal convoluted tubules and the collecting ducts. There it activates transcription so that those cells produce more sodium pumps so that more sodium can be reabsorbed back into the blood. Okay, so again, majority of sodium is reabsorbed by the proximal convoluted tubules, reabsorption, by the ascending loop of Henle, concentration. So what role does aldosterone play? Well, kind of fine-tuning to ensure that the blood has a consistent sodium level. So again, the fall of blood sodium levels activates aldosterone. Aldosterone works directly by activating transcription of sodium pumps in the distal convoluted tubules and the collecting ducts so that more sodium can be removed. Okay, easy, right? Oh no. You ready for the long roadmap of how aldosterone is actually secreted? Near the glomerulus, there is a group of cells known as the juxtaglomerular apparatus. You can see that in the cartoon on the right. These are sodium sensing cells. So if sodium levels drop too far, these cells get excited. And they get excited to release an enzyme. It's not a hormone. It's an enzyme known as renin. Renin converts a prohormone known as angiotensinogen. It cleaves off the big protein part of it to reveal a hormone known as angiotensin 1. Angiotensinogen is released by the liver and is found kind of maintains a low concentration within the blood. The enzyme renin converts angiotensinogen into the hormone angiotensin 1. Angiotensin 1, again in the blood, goes to the lungs. It's part of the circulatory system. There it runs into another enzyme known as angiotensin converting enzyme, also known as ACE. The function of ACE is to cleave off a couple of amino acids to convert angiotensin 1 into angiotensin 2. Again, this takes place in the capillaries of the lungs. Wait, we're not done yet. Finally, angiotensin 2 binds to receptors on the adrenal cortex cells, and it stimulates those cells to produce and release the steroid hormone aldosterone. Right? So aldosterone allows increases the reabsorption of sodium into the blood. But wait, when you increase sodium levels, we have another problem. ADH. ADH senses that elevation in sodium in the blood. It is released to allow water to compensate for that higher sodium levels. So it increases water volume into the blood, which raises blood pressure. Thus, one of the most common drugs that hypertensive or patients with high blood pressure take are ACE inhibitors. ACE inhibitors prevents, somewhere down the line, the production of aldosterone. It prevents an increase in blood sodium levels. It prevents ADH from adding more water to the blood, thus keeps blood pressure lower. The last slide is basically showing the relationship between the aldosterone mechanism and antidiuretic hormone release.